Hello, everyone. I would like to welcome everyone who has joined today for our third and last session under biotechnology category. I am Noeen. I am working as an acting director of event with SCWIST. I am also a chair and lead of Science Symposium. And our today's topic is related to neutralizing the COVID-19 antibodies for detection of antibodies. And our speaker is Anat. I will just go through everything. First, we will give some land acknowledgement. We would like to take a moment and reflect on our connection to the land and thank the traditional guardians of the land that we at SCWIST Vancouver live and work. Vancouver is on the unceded ancestral and traditional lands of Musqueam, Tsleil-Waututh, Seychelles, and Squamish, and indigenous people of Turtle Island. As I already mentioned, today is our last session for biotechnology, and we have Anat with us. She's from Ryerson University, and we have some housekeeping rules. Please keep your mics muted. This event will be video recorded, and later on I will be uploading it on our YouTube SCWIST channel. You can watch it anytime there. And chat box will be monitored. If you have any questions related to the presentation, please type in the chat box and we will take it during our Q&A session. Please note that this Q&A session is very important for us and it is completely dedicated to our audience. So don't hesitate. If you have any questions, just write it down. Agenda is that in the beginning, there will be introduction that I'm giving right now. Then there will be presentation of speaker by Anat for 15 minutes. Then there will be panel discussion. Panel discussion is basically the discussion together with our judges about the research project and other aspects. And at the end, we will have the closing remarks. So we have our two judges with us today here, Dr. Anjana. She is the Director and Strategic Business Consultant at ANRS Consulting Incorporated Institute, Canada. And we have Dr. Brittany, who is tetanus toxoid vaccine manufacturing expert from Sanofi Pasteur. And we have our speaker, Anat from Ryerson University. She will be talking about engineering protein reagents to detect SARS-CoV-2 neutralizing antibodies. This symposium is sponsored by Admare, Bioinnovations, Microsoft Equitas Therapeutics, Northeastern University, Vancouver, Epsilra, Six and Strategy Group, Molly Surgical, and so forth. Some detail about our sponsors. We would like to say thanks to them, and we are really grateful for sponsoring this. From Northeastern University is very famous for biotechnology. The employment rate for biotechnology is 95%. For Admare Bioinnovation, they have started their new program, which is called as Bioinnovations Academy, where you can sharpen your skills about business and about science or benchwork as a research scientist. Then we have Epsilra. Epsilra, they have one news about recently discovered antibody that neutralizes viral variants of COVID-19. We have Equitas Therapeutics. They provided their lipid nanoparticle delivery system, a key element which was used in the development of Pfizer's vaccine. Microsoft, you can check their website. They offer lots of internships and programs in IT. Six and Strategy Group, they are basically the consultancy group. You can contact them if you have any questions about business in life science. And that's all. Here I will stop and I will give floor to our judges, Dr. Brittany and Dr. Anjana, and they can introduce themselves to you. So, Dr. Anjana, if you would like to go first, and then Brittany.
Hello, everyone. I'm Anjana Govindarajan. I'm a trained biological scientist. I have a PhD in biotechnology. Currently, I'm working as a strategic business consultant. I am experienced in establishing collaboration and partnership between industry, academia, and government institutes. I welcome you all to this session. Thank you.
Thanks, Anjana. Brittany.
Hi, everyone. So my name is Brittany Pequegnat. I have my PhD in chemistry from the University of Guelph. I then have a postdoctoral position at Harvard University and Brigham and Women's Hospital. And I am currently the tetanus subject matter expert at Sanofi Pasteur in Toronto.
Thanks, Brittany. So I will give floor to Anat. Anat, you can introduce yourself and then you can share your screen and start your presentation.
Sure. First of all, thank you everyone for coming to our presentation. It is a really big honor. My name is Anat Usatinsky, and I just graduated Ryerson University, a Bachelor's of Science in Biomedical Science yesterday. And I am starting my master's at U of T in Biomedical Engineering in the fall. So this is a really great opportunity for me, and I am really excited to be sharing my project with you. So I'm going to share my screen. I just want to confirm that we can all see my screen. Perfect. Perfect. So once again, thank you everyone for coming. And today I'll be talking about my project, which involves engineering protein reagents for the development of a portable immunoassay to detect SARS-CoV-2 neutralizing antibodies. I'm part of the little lab at Ryerson University, and I'm really excited to be speaking to you all. So we're all familiar with COVID-19 to a certain extent, and as much as we hate to admit it, it has impacted all of our lives. One of the problems of COVID-19 is serology studies, because currently commercially available tests, while they're able to detect antibodies, they're able to tell you if you were infected in the past or are currently infected with COVID-19. They lack the ability to detect the potential for individuals to be reinfected. In addition, those tests are quite resource heavy, they're expensive, and they need an external facility to process the information and require biosafety levels that are quite high. Therefore, we need a way to detect immunity levels in populations. And the reason it is important to detect immunity levels in populations is because decision makers need to know the immunity levels around us to make the proper policies regarding masks, regarding reopening. And the way that we want to detect immunity levels in populations is by focusing on individuals' immunity and by focusing on the individual's immunity to variants of concern. And when I say variants of concern, I mean variants that originated from the UK, from South Africa, and currently the Delta variant, which is the one originated from India. So like I mentioned, the current commercially available tests are quite resource heavy, whereas what we are hoping to address here in the lab is a device that is small, portable, and cheap, and does not require such high levels of safety containment, and is able to detect individuals' immunity and an individual's immunity to different variants of concern, while telling us if the potential of an individual of reinfection with COVID-19. What we chose to do is we chose to focus on this little red protein over here that is seen in the CDC animation. This little protein that is found on the membrane of the virus is called a spike protein, and it has a receptor binding domain, or RBD. And this receptor binding domain could interact with proteins that are found on human epithelial cells, such as the ones found in lungs, called angiotensin converting enzyme 2, or ACE2. And the reason that these two proteins are so important, and the reason that we're focusing on these proteins, is because when the human receptor ACE2 and the spike protein interact, the membrane of the virus is able to fuse with the membrane of the host cell, therefore releasing a genomic content, rendering the host cell now infected. As we know, our body is able to produce antibodies to pathogens that enter the body. A very specific type of antibody that we are focusing on is called a neutralizing antibody. And like the name suggests, this neutralizing antibody neutralizes the virus. And that is because this neutralizing antibody is able to bind to the receptor binding domain that is found in the spike protein. Therefore, the receptor binding domain is unable to interact with the ACE2 receptor on human cells. Thus, the fusion of members is impossible and the virus is rendered unpathogenic. This neutralizing antibody is what we refer to when we're talking about immunity. It is what we hope our body produces after infection, and it is what we hope our body produces after vaccines. Therefore, we believe it is very important to be studying these neutralizing antibody, and therefore we need to find a way to detect this neutralizing antibody in samples in a way that is quantitative. So keeping in mind the interaction between the ACE2, the spike protein, and the inhibition by the neutralizing antibody, we proceeded to be making these proteins over here for our assay. What we did is we used different cells, such as eukaryotic and prokaryotic cells, to produce different variants of the human receptor ACE2, and also produce different variants of the receptor binding domain of the spike protein. And then we used a pull-down assay to purify the protein. And after the proteins were purified, we conjugated biotin onto the proteins and then proceeded to optimize our immunoassay, which we call ELISA. And then after we optimized our ELISA, we moved on to translate this immunoassay onto a paper microfluidic device. So what we're currently working on is we're validating a pre-existing assay that was published by 𝐒𝐏𝐃𝐄𝐑 et al. And 10 et al. And like they did, we are immobilizing human receptor ACE2 to the bottom of wells. We're then adding a biotinylated spike protein. And the biotin is needed on the spike protein because biotin is able to then bind to streptavidin, which we add to solution. And the biotin-streptavidin interaction is often used in labs because the interaction is so strong. And then when streptavidin is found in solution, we're able to observe a colorimetric reaction occur. And when we see a colorimetric reaction occur, then we know that there was successful binding between the receptor binding domain of the spike protein and the human receptor that is found on the bottom of cells. Now, when we're talking about detection of neutralizing antibody in a sample, using this assay, this is what we mean. We still have immobilized human receptor to the bottom of wells. However, if there is neutralizing antibody found in solution, the neutralizing antibody will be the one binding to the spike protein, and therefore the spike protein is unable to bind to the human protein and be washed away. And when we add streptavidin to the solution, there will be no biotin found in the sample. Therefore, the streptavidin will also be washed away because it is unbound, and we'll observe no colorimetric reaction occurring. Now, keeping in mind that when there is color, then we know that there is an interaction occurring, and when there's no color, that indicates the potential presence of a neutralizing antibody. We can talk about how we're translating this assay into a paper microfluidic device. This is a very simple schematic of our device. What we do is we load sample here, we add a running buffer, and then the running buffer diffuses via capillary action through immobilized human receptors on the bottom of the paper microfluidic. And as the sample flows, whatever receptor binding domain is not bound to the neutralizing antibodies will bind to the ACE2 on the bottom of wells, and we'll be able to see a color change in those samples that are bound together. Another thing that we're currently working on now that we validated that the previously published assay does work is we're flipping and creating the reverse assay to address variants of concern of neutralizing antibody of COVID-19. So what we did here is instead of immobilizing the human receptor ACE2 to the bottom of wells, we're now immobilizing the spike protein. And instead of biotinylating the receptor binding domain of the spike protein, we're now doing that to the human protein. So as you can see, we flipped it over. And then similarly to the previous assay, we add streptavidin, and if there's biotin found in solution, the streptavidin will stay in solution. And then we will observe a colorimetric reaction occur, and we can see a color change, indicating that the human receptor and the spike protein were successfully bound. Now, similarly to what I mentioned before, if there is neutralizing antibody found in a sample, it will preferentially bind to the spike protein. Therefore, when we add in a human ACE2 protein, it will not be able to bind and be washed away. And then streptavidin will also be washed away because there is no biotin in solution, and no color change will occur. Now, by immobilizing the spike protein to the bottom of wells, we're able to immobilize different variants on the same assay, allowing us to only use one plasma sample and flow through, and we're able to visualize in a quantitative manner which and how much immunity an individual has and to which type of variant of concern. So now that we established that the assays do work, we wanted to move on to optimizing our ELISAs. We first focused on determining which variant of the receptor binding domain, the shorter version or the longer version, is binding to the human receptor strongly by looking at absorbance readings. And we saw that the shorter version seems to have a stronger binding because we can see higher absorbance readings. So we decided to use the shorter RBD328 mutation. We then moved on to looking at 4 different human ACE2 mutations and variants, and we decided to stick with the pink line over here called mutation 732 because the readings were the most closely together and it had a really nice graph over here. So now that we chose our ACE2, which is a 732 mutation, and our receptor binding domain of the spike protein, which is a shorter version, we're able to move on to testing it with neutralizing antibody. And what we were looking for is to see higher absorbance readings when there is no neutralizing antibody found in solution, and much lower readings when there is added neutralizing antibody. And we did indeed see this curve happening, and we know that our assay therefore works. This is a schematic of the device that we currently have, the prototype. What we do is we add unfiltered and we don't process the sample before, we just straight plasma onto straight blood sample straight to the loading pad. We then add running buffer, and via capillary action, the running buffer diffuses through the immobilized ACE2 on the bottom of wells. And then we're able to observe colorimetric reactions for the receptor binding domain of spike protein that was successfully bound to the immobilized ACE2. And whatever receptor binding domain is bound to neutralizing antibodies gets absorbed in this pad over here. And this is what the device looks like. So the signal we see over here is from the immobilized ACE2 and receptor binding domain that is successfully bound. This sample does not contain any neutralizing antibody. And then we ran further tests, and we saw that we do have a lot more colorimetric change occurring when there is no neutralizing antibody. And as concentration of neutralizing antibody increases, it does what we expected it to do. There's less and less signal with the addition of neutralizing antibody. Now, this device has many significant potential uses. For example, the identification of those people that were previously sick with COVID-19 for passive immunity treatments. And it is also a potential tool that could be used for long-term and short-term vaccine efficacy trials. It also has uses for future disease outbreaks and uses outside of just serology. I would like to thank the Little Lab, Dr. Dustin Little, Dr. Henry. You have been both incredible and always available, and I'm very, very fortunate to be working with you. Our collaborators, Dr. Almastasine, Dr. Rakis, and Dr. Tsai. Our funders, Ryerson University, the COVID-19 Immunity Task Force, the Canadian Institute of Health Research, and Public Health Agency of Canada. And I would like to thank Dr. Noeen, Dr. Ashley, Dr. Brittany, and Dr. Anjana, and the participants for coming today and listening to my talk. And I'm very looking forward to all of your questions.
Okay, thanks, Anat. It was really, really nice presentation. Wonderful. And congratulations for your graduation yesterday. You said you passed yesterday, right?
Yes.
I will give the floor to Anjana and Brittany, so you can start panel discussion.
Sure. I can maybe ask the first question. So first of all, it's very, very interesting technology. It's really nice to see, you know, a faster detection method for us. So something based on your, I guess, colorimetric reaction, how do we know what intensity of the color means that someone has sort of enough immunity, right? Like, do we want to see nothing? Can someone still have some color there and still have full protection? Do we have that kind of information?
So that is actually a fantastic question. And what we're planning to do, which is our next steps, is we're planning on doing a standardized curve. So there are different assays that exist, such as the viral neutralization assay, which, if you're not familiar, requires a high biosafety lab. It requires a lot of equipment. It's quite timely and quite expensive, but it is pretty effective at detecting neutralizing antibodies. So what we could do is we could use the data they have, and if they have any samples that they use that is left over, we are able to create a standardized curve and then see how our device compares to the standardized curve. And then there's a lot of research going on around COVID-19 at the moment. So we're hoping that we can then know and see which levels of immunity protect individuals and which, if asymptomatic people are asymptomatic because they have neutralizing antibody or because of something else. So this is a fantastic question that we are definitely, these are our next steps. We actually got ethics approval now to work with plasma samples of humans. So it is something that we're looking forward to studying.
Okay, perfect. Thank you.
Yeah, wonderful presentation, Anat. Thank you very much for that. I got very much, this kit, you said that this can be used to detect contamination in food. So you're talking about this immunoassay, and can you just explain how can we use this?
Sure. I'll go back to this, for example, this slide over here. So we know that immunoassays in general could be adapted to many different uses. And what we're showing here is but we're able to adapt this immunoassay into a paper microfluidic device, which is light and cheap. So for example, a use could be, like I mentioned, a contamination in food. And let's say I'm looking specifically for 𝐒𝐏𝐃 or E. coli contamination in, let's say, lettuce, then I'm able to create antibodies specifically for this type of E. coli or 𝐒𝐏𝐃, something that is found on the membrane, and immobilize it to the bottom of wells. And then when I add my sample, I'm able to see if there is fluorescence occurring or not occurring, and you adapt the system to have secondary antibodies and whatnot, but our paper microfluidic over here shows that it is possible for us to make these immunoassays on a paper microfluidic and adapt it for various uses.
Wonderful, thank you. So is there any similar kind of portable immunoassay kit available? I mean, to detect any other disease, or I just wanted to know, like, what all available currently.
Sure. So when I try to explain this device to maybe somebody who's not so familiar with microfluidics or not so familiar with molecular biology, I like using a pregnancy test example, because a pregnancy test also uses a paper microfluidic system in which there is immobilized antibodies to the bottom of wells, and they're looking for a specific hormone that is produced by humans when they are pregnant. So yes, so this has been done before. But we're just showing how adaptable this is, and we're portraying further that this paper microfluidic system has many uses.
Thank you.
So as new mutants become, I guess, available, I hate to say it that way, but would we have to then update this technology with the mutant of interest?
Yeah, so that's actually a really great question, and it is one of the reasons, I'll just go to that slide, that we're flipping our device, because it is quite easy for us to adapt this technology. For us in the lab, like, all we need is just to get the plasmid, we need to express and purify it, and then we need to add it to the bottom of wells. But this technology is easily adaptable because we've already shown that we're able to immobilize proteins, and this is just a matter of maybe making the pad longer or wider and creating more channels. for the spike protein of the new variant to be added to. So this device is quite adaptable, and this is what we had in mind when we decided to do the reverse assay. We wanted the ability to adapt it as the virus adapts and to keep up with the times to ensure that we're the most current and we're able to provide the most updated information about the immunity an individual has for various variants.
Mm-hmm. As far as when you were developing your assay, what sort of positive and negative controls did you use?
Yeah, I could go to, for example, I could speak, for example, for our neutralizing antibody test, which I ran another one yesterday, so it's like fresh in my mind. We use positive control to see that, first of all, our immobilized human protein and our spike protein are functional and are able to bind without the presence of neutralizing antibody. So we had a few wells that did not contain any neutralizing antibody, and we were able to see that, yes, they produce color. And then for negative control, for example, we just had, so the buffer we use over here is 3% BSA. So we had wells with just 3% BSA. With immobilized ACE2, we also have various levels of neutralizing antibody in solution. So the beautiful thing about actually the ELISAs that we run in the lab, we're pretty much immediately able to see a colorimetric change. So even without getting the reads off of the plate reader, we're immediately able to see if there is a colorimetric read and how much of it we're able to see. Like you can really see a gradient. So these are just some examples of controls that we use. We see, do we get a read when there is no biotin and streptavidin binded together? And do we get a read when there is a lot of neutralizing antibody?
Okay, thank you.
Any other question, Anjana or Brittany?
How much this is reliable compared to the regular method which we are having? So can you explain how much this is better or reliable?
Yeah, for sure. So from what I understand, antibody tests, especially rapid ones like a paper microfluidic, are only 95% reliable. So it's not the most it's not the most sensitive per se, and there is quite there have been reports of false positives and false negatives, but we are hoping that the use for our device would be for individuals to be checking their immunity for different variants of concern, and we're able to detect presence or lack of presence of neutralizing antibodies in a quick manner, which would allow us for serology testing, and then continuing to further develop this paper microfluidic device, and especially in the context of neutralizing antibodies and antibody detection.
Thank you.
So far from what you've seen with the wild type and the mutant that you've used, it's the same neutralizing antibody for all of them?
So right now, what we're using for our test, this is a mouse neutralizing antibody. And we just recently got ethics approval to use human plasma samples to further test. So we are running tests on different ACE2 variants, because as you could see in this graph over here, we have 4 currently. So we do regularly test different neutralizing antibodies against ACE2. But from what we have seen, the 732 mutant, which is this line over here, has consistently given us more closer together results and more reliable results. So we are sticking to the 732. Also, we need less of the 732 mutant, so we're able to save a bit of money, also make the device cheaper for production uses. So we are using the same neutralizing antibody at the moment, but once we begin using human antibody and human plasma samples, we'd be able to expand our neutralizing antibody tests.
Okay, thank you. And I can ask one more as far as cost of the device. What would you said it's a lower a lower priced device. So what sort of price range would we be talking? And is it something we can deploy, worldwide into, third world type areas?
Yeah, so that is also a really good question. So the price of the device itself, we are still, I don't think we're quite there yet in terms of deciding the price itself. We're still in the research and development stages. But what I can tell you is because we are purifying, expressing, and transfecting and using our own, making our own proteins, the cost of this device is significantly less than if we were to use commercially available proteins. Also, we're currently running trials using a solution called PEI as opposed to using a transfection kit, which is a fraction of the cost of the traditional transfection kits. So we are hopefully trying to cut the costs to less, but from what I've seen, the price, for example, especially right now at the moment with COVID-19 being such a hot topic, the price for the human ACE2 and the spike protein per nanogram is in the thousands, whereas we are able to produce them a lot less than that, in the hundreds levels, and we're able to produce quite a significant amount of protein, and we're hoping to continue producing that and also creating using bigger media as we begin to expand our production, and this will further cut down costs. And to answer the second part of your question about remote use, one of the reasons we are focusing on paper microfluidic devices as opposed to plastic or other forms of microfluidic devices is because paper is relatively cheaper and it is easier to transport. So one of the things we had in mind when designing this device is the ability to do remote testing. And I just do want to say that this device is very user-friendly. It does not require any prior processing of the blood sample. It is similar to a diabetes test in which we just need one drop of blood, so one perk, and one is able to use it. And this is one of the things that we were thinking of because it's not very big, it's 2 inches, and we were thinking about uses in Northern Ontario where there is not a lot of access to facilities that allow for the viral neutralizing assays. So yes, we are hopeful that this can be used in remote locations such as third world countries.
So when can we expect this to be available? Because now we are in pandemic, so now it is very much needed. So when can we expect this? Yeah.
So to answer your question, I was talking to my supervisor and I was talking to him about how, the pandemic is people are saying, oh, it's nearing to an end. But the beautiful thing about our device is, like I mentioned in this slide, it could be used as a potential tool for long-term and short-term vaccine efficacy trials. So what that means is that even though the pandemic is a hot topic right now, and hopefully very soon, we'll forget what Corona even stands for and what SARS is, put it behind us, we'll still be able to use this device to detect the presence or lack thereof of neutralizing antibodies. So while right now it is a hot topic, this tool could be used for many other applications. When would this device become available? We're hoping to start wrapping up the R&D side of things by August, maybe the beginning of September. And then it will be up to my supervisor to decide when they're willing to start talking about production. But personally, I'm hoping sooner rather than later.
Okay, thanks. Brittany, Anjana, do you have any other question?
I'm okay for now.
Anjana?
I'm okay, yeah.
Okay, so here I will take questions from audience, and we asked audience to drop their question in the chat box. This is question and answer session. I will ask questions to Anat, and Brittany and Anjana will evaluate the answers. The first question is, can you tell us more about the application of this type of testing outside of COVID testing, like for other viruses, I would assume.
Yeah, for sure. Again, this is, I'm on this slide perfectly. So now that we have shown that we are able to detect neutralizing antibodies for COVID-19, because this is a paper microfluidic device and because we're producing our own proteins, we would be able to adapt to pretty much any sort of disease outbreak that hopefully won't occur, but could occur. Because we're able to produce the proteins, we know how to add biotin, we know how to use streptavidin, we know how to conjugate the proteins to the paper microfluidic. So all we'd need to know is the neutralizing antibody and then work from there. So this is highly adaptable to other outbreaks aside from COVID-19.
Okay, thanks, Anat. The next question is that why would there be a current need for serology studies? Why would we need to understand immunity levels in the population?
Yeah, that's a wonderful question. So the reason that we need to study serology is because decision makers, such as the ones deciding, especially in Ontario, Brittany, from what I understand, you're at Sanofi, so you're not too far from here. Anjana, you too. We're currently experiencing a lockdown. So decision makers are going to need to understand at what level of immunity the population is. Like, when would it be okay for us to take our masks off? When would we reach the level of herd immunity for us to be able to walk outside, to shop and try on things in person at retail stores? So the reason that we need to understand an individual's immunity is because by understanding the individual's immunity, we're able to see what the population is at. And the reason we need to understand the variants of concern immunity is because then we're able to understand the potential of reinfection of those individuals. So we're not just getting a sense of what the immunity levels are at the moment, at the present, we'll also be able to understand what the immunity levels could potentially be in the future due to variants and due to exposure of different variants and mutations.
Okay, thanks, Anna. The next question is about also what would you say this device can be used for? I think you already answered that, so I'm not taking this question. I will move to the next one, which is what is the advantage of attaching the COVID protein to the bottom of wells and not the human receptor? Really curious about it.
Yeah. So we can go back to this slide over here. So by attaching the different variants to the bottom of wells, we're able to produce a device that runs only one sample and needs only one plasma sample, and is able to diffuse via capillary action to different variants of concern. And like the judges asked, like, can this device be adapted to other variants? Like, yes, it can. We just need to produce the spike protein of that variant. But if we just immobilize ACE2 to the bottom of wells, like we originally did, what we lack the ability to do here is where we can only load one plasma sample and add the buffer containing one type of spike protein. And therefore, we're only able to see immunity for one type of COVID-19. Whereas, like I explained, if we mobilize COVID, like spike protein to the bottom of wells, we're able to add a sample here, and then the buffer would contain the human receptor ACE2, and we're able to run one sample through the device and see immunity levels for that for those different variants and levels for that specific variant.
Okay, thanks, Anna. The next question is how you went about biotinylating the proteins. It is about biotinylation.
Yeah. So I'll go back to this slide over here. So what we did, because we needed to use prokaryotic cells, because we need to express and purify a ligase called BRA. And now this BRA is able to attach biotin to proteins that contain a sequence on them called an AVI tag. And the AVI tag sequence has a very specific part of residue, which is a repetitive lysine residue. and then the BRA attaches the biotin to that specific part of the protein in a specific manner where we want it to be, because this placement of tags is quite important, because we don't want it to be in the active site. We don't want it to interfere with the folding of the protein. So we needed to express and purify BRA and then use it to attach to our proteins. And like I mentioned, this could be done for both the normal assay with the mobilized human receptor and for the reverse assay, because all the protein of interest needs to have is the 𝐉𝐔𝐃 sequence for the biotin to bind to, for the BRA to bind biotin to it in a specific way.
Thanks, Anna. Thanks for this question. Answer. Okay. The next question is, how did you know that the proteins proteins which were expressed are proper? So I think this is what the question is.
Yeah, so for that, I actually have a few more results that I decided to keep just in case somebody asks about the proteins. So if we look at our different SDS gels, and this, for example, we are showing that here's one for the longer version of the spike protein, for example. So we ran an SDS gel and we saw that we got a band at around the size of the expected size of the monomer of the spike protein. So therefore we suspect the protein that we purified and expressed, seeing as it is a proper size, might be the receptor binding domain of the spike protein of the 319 variant. And then to further test not just its size, but its functionality, we performed functional assays. So what we did is we titrated different levels of the receptor binding domain of the spike protein, of different mutations of the spike protein. And then we added the ACE2 to the bottom of wells. And seeing as we saw a concentration-dependent absorbance reading, we deduced that the protein that we have expressed and purified is indeed functioning as it should, because higher levels of the interaction between the spike protein and ACE2 proteins produce higher absorbance readings. So this is how how we did the functional analysis of the proteins.
Yeah, thanks, Anat. The next question is, what types of cells did you use to express the proteins?
So if we go back to this slide, we used BL21 cells and top 10 cells, and we also used XP293 eukaryotic cells. Now, like I said, we needed to express and purify BRA, and this is something that is found in bacteria. So we were able to use BL21 and top 10 cells. Top 10 cells were used for cloning, and BL21 cells were used for a protein expression. So we use prokaryotic cells to express and purify specifically the BRA protein, which I'll remind you is the ligase that we used to add biotin onto our proteins. And then we used XP293 cells, which are eukaryotic cells, to express and purify our ACE2 and the receptor binding domain of the spike protein. And the reason that we actually needed to use eukaryotic cells is because the spike protein has a lot of glycosylation that takes place, and that those post-translational modifications only occur in eukaryotic cells in the Golgi apparatus. Therefore, we couldn't simply use bacterial cells. We needed to now use mammalian cells, which have Golgi apparatus.
Thanks, Anna. Thanks for answering all these questions. I think you did it very well. So, Brittany, if you have any question?
Yeah, I can ask a question. I don't know if you went over this, but so you have your different your variants and your wild type, and you're using the same antibody. Did you see a difference in the amount of antibody required to see the full neutralization for like a similar amount of each of the variants?
So we did see a difference. For example, it's not graphed here, but this graph shows the 732 mutation of 732 mutant of the ACE2, and we're also testing the 732 wild type. And what we saw is that we needed much higher levels of neutralizing antibody in solution for us to begin seeing colorimetric changes, which is why we continued using the 732 mutant. And in addition to that, seeing as we are doing trials with mouse antibody, we are expecting to be using different levels of a human and neutralizing antibody, because from what we understand, the mouse antibody is much more specific, whereas the human antibody will be less specific, so we might need to adjust the levels of neutralizing antibody. But the reason we are using the 732 wild type, like in your question, is because we saw that we need less of the human protein, less of the receptor binding domain protein, and less of the neutralizing antibody to see some sort of trend. And again, this goes back to being cost effective and making sure this device is as affordable as possible. We're trying to use the most optimized reagents.
Mm-hmm. Okay, thank you.
Okay, thanks, Anat. I will ask everyone again for the last time. If you have any questions, please ask now. Drop it in the chat box. About COVID-19 pandemic, you guys have no question? I think I already asked all of them. So let me see. Okay, I don't see any other question in the chat now. We covered pretty much every question. Okay. Anjana, Brittany, you both are good?
I'm good.
Okay.
Yeah, thank you.
Okay. Okay, so let's then wind up, right? So Anat, you can stop sharing your screen. Okay. And here I would like to say thanks to Anat for presenting her wonderful research with us. And I would like to say thanks to our judges, Brittany and Anjana. It was our last session for biotechnology. And I must say that I am really, really grateful for their time. I know that both of you are very busy, but I also know that both of you are very well experienced in this field. And that was the reason that we invited both of you. And it was a really good experience of running these sessions. I would like to say thanks to all of our attendees who came today to support Anat and to support our sessions, and all our sponsors for sure. I will now share my screen and I will say some few things. Anat, do you have any question for me, or you are all good, right?
I'm all good. I just want to say thank you everyone for coming. Thank you, Anjana and Brittany, and Noeen, everyone else who helped organize this conference. This has been a really great experience, and it's really great seeing the support in the chat.
Okay, thanks, Anat. Brittany and Anjana, would you like to say something as closing remarks?
Sure, I can go first. Yeah, this was a great presentation. Obviously, this sort of thing is very close to my heart in my field, and with the new initiatives at Sanofi moving forwards with looking at flu and pandemic-like instances. This is just very interesting, and I think it has some great applications, not only for long-term and short-term immunity, but looking into if boosters are needed and how quickly we need to get boosters on the market. So this is something that can be very valuable. So I thank you for sharing, and this was a great third session for the biotechnology field. So thank you.
Thanks, Anat. It was a wonderful presentation, and I'm very happy to see young scientists here. So you completed your undergrad, and now you are entering a master's, and I'm hoping that you will be getting a lot more good research and good products to Canadian economy. Yeah, once again, thank you for sharing your work with us. And I also thank SCWIST for giving this opportunity to learn about these new research in different fields of biotechnology. So biotechnology is not just one, right? So it includes food, everything. So you have covered everything. Yeah, I once again thank SCWIST, and I also thank Anat for today's presentation. Thank you.
Thanks, Anjana. Thanks, Anat, and thanks, Brittany, once again. And I will now share my screen. I know you can hear like message notices sound in background, but let's just ignore it, right? So, okay. Yeah, so I will start from here.
Noeen, did we lose you?
It looks like we may have lost Noeen. We can give her a second to see if she comes back, but otherwise I can give a quick summary of what I believe she was going to talk about.
Go ahead, Brittany, I think, because just in observance of the time.
Yeah, that's true. So I don't have my slides with me right now, but I would just like to let everyone know that if you want to follow along with the upcoming Science Symposium events, you can head on over to the SCWIST Eventbrite, and you can see them all there and register. You can also go to the SCWIST website, and you'll be able to find all of our events on there. Oh, and I think we just have Noeen coming back in. Hi, Noeen.
Hi.
I was just letting everyone know that they can head to Eventbrite or the SCWIST website to find all the upcoming symposium events. Yeah.
Thanks, Ashley. You made this job easier for me. I think my internet again kicked me out, actually. So I was just thinking, but I just near the closing remarks and I am out. And I was just thinking, thank God you're recording this session. So yeah, thanks everyone. I think here we can go, we can leave, we can close the session. And Ashley has already talked about our future events, future sessions. Next sessions will be for the category drug discovery. We will have first session next week. And that's it. Have a good day, good evening, good afternoon, and enjoy sunny weather, snow, rain, whatever you have. So take care. Bye.
Thank you, everyone.
Bye.
Thank you.